This will be our 19th Chestnut Chat of 2020. Of course, we started these back in the spring when everything went virtual, and we'd never really done this kind of a series before. And I think since then, most organizations have started similar webinar series. We've had over 1,000 people, unique visitors to our Chestnut Chats. We average about 100 viewers on our chats every time, and our chats tend to go about an hour and a half or a little over every time. So we'll probably go about an hour and a half again today. Again, this is our last chat of the year, and we started our Chestnut Chat series with an intro, me, Lisa, and Jared. We talked a little bit about what we're doing, why we got involved in TACF, why we love Chestnut, and then answered all your questions. And so we want to end like that too. So our session today is a town hall. We're going to answer all your questions. We had a bit over 60 questions submitted as part of the call for questions ahead of time. Lisa is going to be moderating the session today and feeding us some questions. And you've got the 5 panelists here. I'm going to introduce myself before I turn it over to Lisa, and then maybe we'll have the rest of the folks introduce themselves. I'm Sarah Fitzsimmons. I'm the Director of Restoration, and I've been your mostly host for most of these Chestnut Chats, and I work out of our Penn State office, and I've worked with the American Chestnut Foundation for almost 20 years now, starting as an intern in 2000 with the Pennsylvania, New Jersey chapter. So with that, again, I want to thank you guys for coming, for submitting questions, for being so interested in our work, and I'll turn it over to Lisa.
Thank you, Sarah. Again, I want to add my thanks to hers for this happy surprise, in which was a very challenging year, is the Chestnut Chat. We kind of just started it off kind of informally to be able to reach out to you and increase engagement when we're all separated and isolated, and it's just been such a great success. We are moving to 1 Chestnut Chat a month starting next month, and we'll get all those announcements out. The first one will be January 15th at the same time, 11:30 Eastern Standard Time. So with that, I won't take too much time because there is a lot of interest. Chestnutters are a curious bunch, and that is awesome. That's why we are so lucky to have a volunteer workforce, and the curiosity and the interest and sincerity of purpose that all of you have to join us is just really inspiring to me personally. So we do, Sarah mentioned that we'll be prioritizing questions that are unique or which, a lot of folks, had the same ones. If we don't get to your question, be sure to check out previous Chestnut Chats. There very well could be answers in there to your questions, but we'll really do our best to follow up via email if you put things in the chat or in the questions. So the opening question is for the entire panel, but maybe we should do a real quick introduction first. Tom, why don't you start? Regional Science Coordinator for 2, not 1, but 2 territories right now. Tom, take it away.
Hi, everybody. My name is Tom Saielli. I am currently the Mid-Atlantic Regional Science Coordinator, and I'm covering the Southern region. I used to be the Southern Regional Science Coordinator for TACF. Before that, from 2008 to 2012, I worked with Kendra Collins up in New England. So I've had a chance to get around a little bit, and I've seen just about all the orchards and met a lot of the people. And so right now I'm in Charlottesville, Virginia, and loving chestnut work here.
Thanks, Tom. Kendra, you want to go?
Sure. I'm Kendra Collins. I'm the science coordinator for New England, based out of Burlington, Vermont, at the University of Vermont, and in partnership with the U.S. Forest Service. I started working with chestnut in grad school in 2005 and haven't looked back since.
Jared?
Hey, I'm Jared. I've been with the foundation since 2015, and I'm the director of science. And basically my job is been a lot of data analysis for the foundation and building collaboration with university partners, and basically just helping us try to understand the resistance and select the best parents and work smarter, not harder.
Thanks, Jared. Andy.
Hi, Andy Newhouse. I work at the SUNY College of Environmental Science and Forestry in Syracuse, New York. So I'm the only one here, I guess, not on staff with the Chestnut Foundation, but very pleased to work closely with all of them. So thanks. And I've been working on chestnuts at ESF for, I think, 11 years now. Our team has developed the transgenic American chestnut, and I'm specifically focusing on the regulatory process. So we'll get to some questions about that.
Thanks, Andy. And before we get started, I wanted to do a shout out to our perfect attendance Chestnut Chat attendee, Carl from Pennsylvania. Thank you for joining all 19 Chestnut Chats. You're the only one in the foundation or our community that's done that. So thanks for that loyalty. So the opening question, Louise and Mike Alcott, wonderful volunteers in Pennsylvania. As the scientist and writer Isaac Asimov once said, the most exciting phrase to hear in science, the one that heralds new discoveries, is not eureka, but rather, that's funny. So what is making you all say, that's funny these days? And how does that translate to your priorities for new research? So esteemed panelists, who would like to take a shot at that first?
I'll go first if y'all don't mind. The biggest, it's not just this year, but I think it's been heightened this year with the public comment period and kind of thinking about the question of is the transgenic tree a plant pest risk? But that was the question that we had to answer with regard to the public comment for USDA APHIS. And what's been making me go, most is why a lot of these older hybrid plantings that are 100+ years old or Chinese chestnut plantations are not naturally reproducing underneath. And I've been curious about that for some time. I keep wondering about that, but as I look at it more and more, it makes me more and more curious. This past year, I was able to visit Lesane State Forest with Tom Scieli outside of Charlottesville. And what a fascinating and inspiring place, a place with 10,000 hybrids that are 50 years old, and you see little to no reproduction in the understory. So, from a restoration standpoint, that's really interesting, very intriguing, and is the question that I'm mostly going, why is that these days?
I'm going to follow up, Sarah, because I think you saying that is what's funny to me, because what I was going to say is funny was also came from walking around Lesane State Forest. So Lesane State Forest, it's the Virginia Department of Forestry's breeding program. It's similar to ours, but different material, and their approach has been different. And it's been more of a planting the trees and letting them sort of emerge on their own and develop into timber type trees and sort of fight it out. And so we've been trying to do a lot of assessing of those trees. And this summer I had a that's funny moment when we noticed that the biggest, best trees that were the tallest and most dominant and the most timber type were the ones that actually had slightly more Chinese traits than siblings from the same families that had more American traits. And my hypothesis was that the more Chinese trees were dealing with blight better than the American type trees that were struggling. And so, it led me to believe that we really have to assess our material really well, which we're doing, and use the best tools possible, which we now have, or at least we're using the best tools today to be careful with our selections. But it was really an interesting thing to find.
One thing I've been thinking a lot about is, how do we do our selections based on blight resistant or blight selections for blight resistance? And we've been evaluating seedlings for resistance. We also inoculate smaller trees in the field, like at age 3, so they're like this big around. And we have been looking at the trees, and we look at the cankers like 12 months after inoculation. And then we also been looking at the long-term resistance of these trees. So like 2 or more years after they're inoculated, does the main stem survive, et cetera. And we've genotyped a lot of the trees. So we've assessed the DNA. And what it looks like is that there might be, blight resistance actually might be a complex phenomenon where different kinds of resistance mechanisms come on at different stages of the infection of the tree. So early on in the infection, it might be important that metabolites in the stem, things that minimize the growth of the fungus, or oxalic acid detoxification, like with the OXO gene, might be very important to limit the growth of the fungus. But then later on, the tree starts to form physical barriers to the fungal infection, and that ability to form those barriers, the thickness of those barriers, the rate that they're formed might be an important component of resistance. So resistance is actually a complex phenomenon involving different time scales. And so I think we have a lot to learn in terms of what's going on at different time points along this as we're measuring resistance.
Kendra or Andy, you want to add to this?
Yeah, I can add a quick one because I think the points that Sarah and Jared just brought up are both pretty good ones for our science program. This is a little more ancillary, but I think it's been kind of interesting this year. There's a chestnut specific bee, Andrena rennie, that has been sort of rediscovered. I think it was found last year in Connecticut at the Ag Station, and a few more folks have become interested and started looking for it. And the story with it is that it's a chestnut specific pollinator. And most of our understanding of chestnut specific species is that they have been lost along with the loss of chestnut. I mean, we consider chestnut functionally extinct because it doesn't typically flower and reproduce sexually. So I'm really curious just to see where that research goes. I don't think it's something that, you know, we will be able to necessarily put a lot of our own resources into, but certainly we can partner with folks who are looking at this pollinator. And I'm just curious what has it been feeding on other than American chestnut? Because if it is so chestnut specific, you know, a lot of native pollinator species don't necessarily go after the exotic ones that we bring in, so like Chinese or Japanese chestnut. So I'll be interested to sort of just see how this story unfolds and what we can learn.
For me, I think a lot of what has been making me go is based on what we've been learning recently about genomes, whole plant genomes, and different methods that we can use to modify genomes. And that, of course, from our work is can be intentional and very specific with transgenics, but also with hybridization, with traditional breeding. That's another way of changing genomes and affecting plants. And as not we specifically, as scientists have been sequencing genomes and comparing genomes between plants, and the technology has caught up to the point where we can actually look at and compare whole genomes that have been modified in different ways. Hybridization can result in large changes in many different places. And you might assume, I would have assumed that large changes in many different places and lots of chromosomes would mess up a plant or something, but we see lots of healthy hybrid plants, chestnuts and otherwise. And so that's interesting to me. And we just found out that a paper was accepted kind of talking about that in more detail and different scales of changes by different types, and thinking about how you might assess risks and so on. So hopefully you'll be able to read more about that soon.
Great answers, everybody. I'll go on to the next category, which is the public comment period that we just went through for the USDA APHIS. Sarah mentioned it earlier. From Brad Johnson, he's an educator. What was the final breakdown of positive versus critical for the public comment period? Are people starting to accept the safety of this technology? I'm going to just rattle off some fun facts that we did some content analysis of the 4,337 comments that were filed. We had a very robust volunteer task force with not only volunteers, but some of the staff from ESF. And of those 4,000 plus, 2,694 were positive or supportive, that's 62%. 1,589 were negative, 37%, and about 1% were neutral, about 54 comments. And we were really thrilled with the goodwill that was generated for the most part by this process. We got incredible engagement from the scientific community, professional scientists particularly. 71 universities were represented in the comments. Over 100 organizations were represented, including the Nature Conservancy, which is my alma mater, Society of American Foresters, and Environmental Defense Fund, to name a few. So, you know, we're really thrilled with the response. And for those of you in the audience that sent in a comment, took the time, we really, really appreciate it. So there will be future public comments periods that we need to stay on top of, not only for the USDA APHIS, but for the Environmental Protection Agency and perhaps the FDA. But I am going to flip the rest of that comment over to Andy Newhouse, because from David Guazi. Hi, David. Good to see you on here. Christopher Kernan, Robert Carr, Paul Anderson, and Stephen McGill. They have a 3-part question. If you forget all 3 parts, Andy, I'll repeat them. Any updates since the public comment period ended? What's the status of the application with the USDA? And has Darling 58 been approved yet? And when will that happen?
Sure, I can kind of intertwine all of those, I think. I guess to start with the last one, no, Darling 58 has not been approved yet, but it's in process. And so the public comment period was basically the first part of the USDA's assessment of the Darling 58. And the next part, or the current part, that they're writing up a document called a plant pest risk assessment, and they incorporate the public comments. The whole public comment period was part of that, was informing the plant pest risk assessment. And in parallel to that, I guess a different department is writing up a different document called an environmental impact statement. So we've been told those 2 documents might take about 6 months. And so realistically, with the way everything has been going this year, I'd give it about a year, or we don't know how long it'll take. But on the scale of months to a year, as they're preparing those 2 documents, once those 2 documents are ready in draft form, there will be another public comment period kind of in response to those 2. It'll be a shorter period. But those are the updates that they're working on, preparing those 2 documents. And then I think that's enough detail for that. It's in progress.
Just to follow up, Andy, Clark Beebe asked how does this compare, the amount of comments, 4,600 plus, with other USDA APHIS comment periods? Do you know that? I'm not sure. I think it's pretty robust, but you might know that better than me. Mm-hmm.
It's hard to decide what to compare it to, because comparing it to a corn crop or something is just so different. It's hard to decide. One other interesting one, a unique type of plant that's not a typical agricultural crop, I guess, is cotton, a modified cotton, where the seed was modified to make it edible. So yeah, cotton is a big crop and the fiber is harvested, but the cotton seeds have this toxin that they're otherwise nutritionally pretty good, except for this one toxin. And so a university group like ours modified cotton seeds to remove this toxin to make seeds edible, which would be great. You know, we could harvest both the fiber and a seed. And so that's kind of another example of something, a unique application of biotechnology, I guess. And I think that their open comment generated something like 50 comments, less than 100. And so it's again, pretty different. It's hard to compare it to chestnut used for restoration. But yes, we certainly saw a lot of comments, a lot of interest. There have absolutely been other crops that have generated more comments, but this is pretty unique.
Thanks, Andy. We're going to turn over to another topic, chestnut types, and this is one aimed at Tom Scielli. It's from Rod Walker. I keep finding articles about people growing more and more commercial orchards of chestnuts using the Dunstan chestnut. What is this chestnut and how does it compare to what TACF is producing?
Right. So the Dunstan chestnut, just a little background on that, is something that's propagated at a nursery in northern Florida at Chestnut Hill Nursery. And this is work that started back in the 80s when a guy by the name, what's his name? Mr. Dunstan, anyway, found some American chestnuts, I think in Ohio, and he grafted them onto Chinese rootstock and then started to his own sort of hybrid breeding program with that initial find. And he produced a tree that is a hybrid, but this is a hybrid that is indistinguishable from Chinese chestnut. I mean, it's a little bit distinguishable. When I look at a Dunstan, I can tell it's a Dunstan because it's not a perfect Chinese chestnut, but it's mostly Chinese chestnut. So probably that original F1 was crossed to a Chinese chestnut, which is great because the Dunstan chestnuts are highly blight resistant and can tolerate a lot of soil types similar to other Chinese chestnuts. So people who grow Dunstan chestnuts are growing an essentially Chinese chestnut type of a tree. It's a hybrid, but it's very Chinesey, and the nuts are especially Chinesey. If you look at the nuts from Dunstan chestnuts, they look very similar to Chinese chestnuts. So that's what the Dunstan chestnut is. And yeah, and Sarah's pointing out that there was an article written about it in the magazine, and maybe we can get that out and put it at the, put it out there for folks to be able to read more about Dunstan chestnuts. But anyway, I think Dunstan chestnuts are nice trees, but I also think Chinese chestnuts are nice trees. They produce large edible nuts, but it's not going to be a tree that we can put back in the forest because it's so Chinesey. It probably won't be ecologically fit to replace American chestnut. But it could be used for other things. People can plant them on their property. You can maybe use it for a food plot, but I'm concerned about having a large Chinese chestnut seed for wildlife as opposed to the American chestnut seed, which is actually quite small. And a lot of animals that would consume the seeds probably can't handle a large Chinese type seed like the Dunstan chestnut seed. So that would have to be taken into consideration. Getting to the question though, about people who are using these commercially. I don't have a problem with that, except, well, I do have two issues with that. Oftentimes people who grow Dunstans commercially will sell them as an American type chestnut. And as I just pointed out, they're not an American type chestnut, they're a Chinese chestnut. So I would like to see a little more, clarity there. If you're gonna sell a Dunstan chestnut, don't sell it as an American chestnut. And then if you're growing chestnuts commercially, there are some great cultivars out there. And folks up in Michigan who work with the Northern Nut Growers Association could help anybody with looking at the cultivars that are available for chestnut growers. There's some great Chinese chestnuts that were actually bred to be a nice cultivar, meaning they're delicious and they're easy to peel. And Dunstans, in my opinion, are very difficult to peel. They're like a lot of other Chinese chestnuts. They're not bad, but they're not delicious, hard to peel. I'd say if you're going to do this commercially, you should get in touch with other folks who are growing chestnuts commercially, who work with some of the varieties that were bred to be a good commercially grown seed, and then be honest about what it is. It's not an American chestnut, but it's a great seed. So I'm not thrilled with folks who grow Dunstans commercially. I don't think they're a great seed to eat, and I don't think it's fair to pass them off as an American. That said, TACF isn't going after these little mom and pop farmers who grow Dunstans or whatever else they want to grow. We just sort of leave it up to the buyer to decide what they want to buy and eat.
I just wanted to add, I was looking at,
Thanks, Tom.
we had done some DNA analysis with Dunstans, and it looks like they have more than 90% Chinese ancestry. So that's just like one seedling. So it's not like all Dunstans, but they are more Chinese. The DNA confirmed that they're more Chinese than they are American, which is not the same as generating backcross trees with American chestnut, which are more American than Chinese. Even if they do have some of our trees have 25% or more of their genome from Chinese chestnut. It's a big difference between a backcross tree and a Dunstan tree.
Thanks, you two. Just a reminder, if you have a question, put it please in the Q&A, and the panelists will be monitoring that and may be able to type the answer. We are giving priority to those of you that sent in questions ahead of time. We're not ignoring you on the chat or the Q&A, but we'll get to them as best we can with the great interest that we had in a number of questions. So going to go on to Jared from Alex Baldwin. Some oak trees get the blight, but most U.S. species aren't affected. Why don't the hyphae of Cryphonectria parasitica penetrate any other tree's bark enough to destroy it completely, but affect the American chestnut so detrimentally?
Yeah, oaks, Cryphonectria does live on oak trees, like especially scarlet oak and other trees that coexist with American chestnut. They get small lesions on the bark from Cryphonectria infection, but the fungus does not penetrate deeply into the bark like it does with American chestnut. And why that happens, what's the difference between oak trees and American chestnut trees? I don't know the answer. And to be able to answer that question, even with Chinese and American, comparing the Chinese and American, what we do is we cross the trees, we make hybrids of them, and then we look at which trees are more resistant and which trees are less resistant. And we compare the genomes and we say these regions of the genome are associated with resistance. You can't make a cross between an oak tree and a chestnut tree to do that kind of analysis, the susceptible American chestnut with an oak tree. So I don't know how you would answer that question. Honestly, like why is the oak resistant and the chestnut susceptible? I don't know. Andy, I don't know if you have any more ideas on that.
Other than looking at some of the parts of the infection process that we understand, like certain enzymes that the fungus uses to attack chestnut tissues, and whether those are present at similar levels or whether there are compounds that might interfere with them in other species. But right, as far as comparing specific mechanisms between different unrelated species, that'd be tough.
And I think, part of that question too was like, why doesn't Cryphonectria attack ash or, ailanthus or whatever it is? And I mean, it's the same reason that, you don't necessarily have diseases that attack cows and cats and, things like that attack people. Sometimes things cross. I mean, coronavirus is an example of that, right? You've got something that has crossed the animal person barrier, and sometimes that happens in plant diseases too, but it's a whole mess of host pathogen recognition, that the pathogen recognizes the tree as a host and the host recognizes the pathogen as a pathogen, and you've got a whole slew of things that occur just to try and figure out who are you. Wait, who are you? Can I eat you? And that all has to happen first before you can actually have some sort of pathogenesis. So it's a very complicated thing. I know I can't offer any specifics, but I tend to like to personify and anthropomorphize things. So I don't know if that's helpful.
Bruce Levine had a helpful comment in the Q&A along the same lines that the fungus has probably evolved somehow to be specific on chestnut, and we don't know why, but that's the way pathogens or saprophytes tend to work.
And one thing that chestnut has is a lot of tannin in the bark, and the fungus lives off of it derives energy from the tannins in the bark. So maybe the specific types of tannins in American chestnut may be tastier to the fungus. And like Sarah said, like oaks, like they might have a lot of genes involved in pathogen recognition where they see the pathogen and they immediately are able to say like, oh, we've been infected by this and let's start to mount a defense response. And maybe that's lacking. The ability to see that they've been infected is lacking in American chestnut as compared to Chinese chestnuts or oak.
Great conversation there. This one's for you, Sarah, from Frank Skalek. And all of you, if I botch your last name pronunciations, forgive me. At one time, Fred Hubbard told me if you have enough trees, you would eventually get hypervirulence to develop. Is this happening? Is hypervirulence expanding?
Good question. And thanks for submitting that ahead of time. I did consult with Mark Double a little bit about that too. And from my personal experience, no, hypervirulence is not expanding. And that's Mark's observation as well. So most of the hypervirulence that is in the US has been introduced from Europe. there are some examples of wild hypervirulence that are out there. It's few and far between, and that would neither type, the introduced version nor the what appears to be naturally or wildly derived, are spreading. Unfortunately, it's still a mystery as to why they're not spreading here and why it does spread more readily in Europe. But no, unfortunately, and we're still looking at it.
Yeah, one of the things I've also heard is that there's been multiple introductions of Cryphonectria into the US, and that because it's a more diverse collection of fungus versus in Europe when there was like more limited introduction, there's a lot more, this diversity actually inhibits the transmission of the hypoviruses between strains of the fungus. So because they're not able to move this virus around as readily, it doesn't spread. And whereas in Europe, because there's like a much more limited diversity, it's able to spread in the fungal population easier.
Great. And for those of you who don't know Mark Double, he is retired now, right, Sarah? But led the effort for hypervirulence research here, along with Bill Nuss. Is that right? Am I getting his name right?
Don Nuss.
Don Nuss, thank you. Over in West Virginia University. So great researchers there doing really great work. This is an overall question about TACF, and I'm going to flip it over to Kendra from Bill Loftus. Is there anything to see on the farm in Virginia, meaning the Meadowview Research Farm? And if so, are tours available?
Nothing there. No, there's a lot of chestnut trees, an awful lot of chestnut plantings at various stages of growth, some different collections. There's actually 4 different farms that are all kind of grouped under the heading of Meadowview. Tours aren't something that we formally offer, but if you're in the area and you're interested and there's not a global pandemic happening, you can give the farm a call, and they're usually, as long as they have time, they're happy to show folks around. Certainly if you give them a heads up ahead of time, that's helpful. Just showing up in the middle of their workday is probably not the best way to get a tour or to get their goodwill towards that effort. Dan McKinnon, I saw, is actually on as an attendee. He's our operations manager at Meadowview. So if he has anything to add, Dan, I welcome you to throw that in the chat. But yeah, it's a really interesting place to visit if you have the time and the staff has the time to show you around.
Great, thanks, Kendra. Back to transgenics, to Sarah Fitzsimmons. This is from Lawrence Jones from Yatesville, New York. How do I prioritize my limited planting space? Do I plant wild type American chestnuts, which will just die from blight? Do I save those spots for transgenic trees?
I get that similar question a lot. And I think it really depends on your goals as a landowner. I think that it is always a good idea to plant wild type American chestnuts. Even if they die of blight, you can replace that easily. Because they die of the blight, you can replace it easily. They don't typically get large enough to where you have to worry about, quote unquote, saving the space for something else. So I wouldn't worry necessarily. I think it's, like I said, it's always a good idea to plant wild type. I can't think of a reason in this respect to not go ahead and do that. And we're going to talk a little bit about distribution a little bit. I'm sure what you all, what many of you got on here about was to find out when you can get some transgenic trees or some advanced, blight resistant backcross trees. And, one of the best ways that we're going to be able to get material out with ESF is, and Andy's going to touch on this, is pollen. And if you have American chestnuts already established and they're flowering, you'll be able to get pollen more easily than a seed. And so if you plant wild type Americans or even hybrids and they're flowering, you're going to be able to breed on that. The sooner you do that, the sooner they have flowers, the sooner you'll be able to do breeding. So, I wouldn't worry about saving space at this point. I would go ahead and plant something, figure out how to plant them, figure out what, if your site is good enough to support chestnut, that's going to be your priority for right now, looking to the future to plant something, better in the long term.
Great, thanks, Sarah. All right, this one's for you, Kendra, from David Stout. Is there any plan to restore the American elm or white ash using a method similar to that used at ESF?
Yeah, so we'll start with American elm. Yes, ESF actually has been working on American elm. Andy informed me this morning that he did his master's work on that. And Allison in their lab is sort of taken over that project for the time being. So there is a similar effort on elm. There's also been breeding work and cultivar selection work with elm that's ongoing. And with ash, it's a little bit farther behind. We haven't had emerald ash borer plaguing us for nearly as long as Dutch elm disease. There is some breeding work going on with ash, some real preliminary stuff with the U.S. Forest Service. I'm not aware of any transgenic research with elm. That we're still kind of at the front lines of looking at what we can do with the pest. Conserving the germplasm that's out there and kind of figuring out what the best approach is going to be. I think kind of like the early days with chestnut blight, you throw a lot of things at the problem and see what sticks. And if there is an opportunity for a transgenic solution, I'm sure someone will take that up sooner than later. Andy, if you have anything to add to that, feel free. I think this is going to get tossed to you in a minute here anyway.
Sure. On the ash, I would just add that there has been some preliminary work on transgenic ash, but not really actively trying to plant it out or do outdoor field trials even, I don't think, at this point.
The ash people are very advanced in the genomics realm. So what they've done is compared the resistant susceptible species, and they're not like all Asian species are not all resistant to the emerald ash borer, and not all North American are equally susceptible. And so they're looking at genes that look to be like conserved in all the resistant species across this tree, the tree of life. However many ash species. So they've identified 30, 40 genes, I think, that looked like they were associated with only the resistant trees. And then they were talking about doing gene editing on that. So it actually advanced more probably than in chestnut on the genomics front and understanding the mechanisms involved.
And I think part of that is because, as I understand it, there are a very small percent, but some ash trees that actually have substantial resistance to the emerald ash borer, or at least some sort, like people kind of hoped was the case with chestnut, but is rarely very much or very significant resistance. And so that has probably informed this genomic work that Jared has talked about.
Great, thank you. And Andy, you're on the hot seat for several questions in a row, and I'll just guide you through them if you don't mind. And by the way, Dan McKinnon, our Director of Farm Operations, did put in the chat how you can visit in the future. And we are in an overabundance of caution, not having visitors until the COVID danger is diminished. And so we're looking at probably next summer. Oh, it only went to the panelists, sorry. So I don't know how to get it over into the panelists. We'll figure it out. So Andy from Robbie Shaw. Hey, Robbie, does the transgenic program rely on the regenerating roots, or is it solely new DNA clipped seedlings that propagate future resistant trees?
Good question. And I think if I'm understanding the question correctly, we cannot modify an existing tree. Like if a tree is standing there, we can't add the new DNA to the entire tree. That's really not how our process works. So we need to generate new trees that have the transgene. And I hope that answers the question. It's not exactly DNA clipped, it's a new piece of DNA in this case, but hopefully that makes sense.
Great, thanks. From Michael Gaffey from Old Lyme, Connecticut. How many fertile darling chestnuts are harvested from current test plots each fall?
Well, we're still expanding that and still working on the process, but over the last couple of years, we've harvested on the scale of a couple of thousand chestnuts from transgenic crosses.
And from Mimai, I hope I'm pronouncing that correctly. How tall will these trees grow and how big will the nuts be?
Just the same as other American chestnuts. the differences we're seeing might be really small, maybe a few % differences in growth rates. But we don't have any reason to think that the transgene will change the overall height, and we definitely haven't seen any differences in nut size between transgenic and non-transgenic. Those are very similar. We've tracked that quite a bit.
Very good. And then several questions from Paul Anderson. How will we be able to test nuts in the future for presence of OXO at our homes?
Good question, and it will be really relevant. The simplest answer, and maybe most casual if you're really patient, is to just let everything grow and see what gets hit by blight worst. But more specific than that, and especially if you want to be intentional about crosses or transplanting or whatever, we have an assay that's very easy to do in the lab, where you can take a leaf disc, like a little punch out of a chestnut leaf, or if you're really careful, you can take a slice or a core out of a chestnut. You run the risk of damaging the nut that way, but it is possible. And then drop that little piece of tissue in a tube or in a pair of tubes, and then look for a color change. And if it turns black, then the transgene is there and active. That's easy to do in the lab. It's not easy to make those solutions at home, but we would hope to plan to share those tubes like a pre-made kit of some sort. And I don't know how that would work as far as distribution or costs or whatever, but that is something that we can send. We can send this little pair of tubes to you to take a hole punch out of a leaf and drop the tissue in the tube and check it the next day. And if it changes color, then it's transgenic. And if not, then it's not.
Okay. Then he asks, there seems to be 2 generations of transgenic trees. How is the D58, Darling 58, different? Is there an additional accelerator gene? Please explain.
I'll do my best. We've probably used the word generations in a couple of different ways. And one is just breeding generations. So Darling 58 was a clonal line, was the original tree that we generated in the lab, but we were able to propagate those. So we have lots of original Darling 58 trees. We grow those up and collect pollen, and then use that pollen to pollinate wild type trees. And then those offspring are a second generation. They're still Darling 58 because the ones that inherit the transgene have the same transgene in the same place in the genome. But that's what we would call the T1, or the first offspring generation from a transgenic tree. And then we've successively collected pollen and bred them. We're now working on a third generation of Darling 58 offspring, but we would still call those Darling 58 because it wasn't a separate transformation. The gene is the same gene in the same place in the genome. Another way we've used generations, though, is to talk about kind of like you'd say generations of iPhones or something, like the first one that gets developed, and then the second one, you optimize it and change some things and release the second generation. The way we would use that term, we wouldn't say Darling 58 for a different generation. So several years ago, we were testing a different, what we would call a different event called Darling 4. It also had the same transgene, but it was there were some differences with the sequences surrounding the transgene. It was a different event. It was inserted in a different place in a different background line. So that was, we probably used the term generation. It was a different generation of transgenic trees, but it's not Darling 58. That we are not seeking non-regulated status for that. It's separate. And then we would also talk about future generations. Like we would hope to further optimize eventually, make trees that might be even better than Darling 58. And that we should probably come up with a better term than generation, but to avoid confusion with offspring generations. But that's in process too.
Well, that segues into what further work is being done on the Darling 58.
We're continuing long-term ecological tests as many have been started. We have done as many tests as we can on trees the size that we have, but of course there are many interesting tests yet to be done with bigger trees and longer term tests and larger scale tests, and those have been started. And then I guess to kind of continue the question a little bit beyond the way it was asked, further work with, if I can say, oxalate oxidase transgenic chestnuts, I guess, if we use the same gene, but if we insert it with a different control sequence, I guess I'd say, it's called a promoter more specifically, where if we could target the expression of the transgene, so it's only active or on just under the bark where the Cryphonectria attacks the trees, that would probably be even better. The Darling 58 expresses the transgene kind of everywhere in the tree, and that seems to work at protecting the trees. But if we could optimize it and kind of target that expression to either tissues where the Cryphonectria is attacking the tree, or specifically in response to the pathogen infection, that might be even better. And we're looking at that.
Great. And finally, what is the feeling on the Darling 58? Is it better than Chinese for resistance? And what can be expected for survival percentage? And one quick follow-up from the chat. Did I hear you right? The transgenic tree may have a slower growth rate. So better than Chinese for resistance, survival percentage, and growth rate.
I don't think I can say better than Chinese. In some of our really early seedling tests, the transgenic trees survive, a higher proportion of transgenic trees survive compared to the Chinese seedlings, but those are potted seedlings that are this big under controlled conditions. So in a few years, when we have older transgenic trees, and we're talking about, I just yesterday was talking with Jared about how to kind of plan these experiments to include Chinese trees and hybrids and backcross trees, and really be able to get some longer term data. I think we'll be able to answer that question better. I would say, based on what we know so far, both can be effective at enhancing blight resistance. I couldn't make a claim on better right now. What can be expected for survival percentage? Good. I don't want to put a number on it right now. We'll know better in a few years when we get older trees. And as far as the growth rates, we are still looking at preliminary data, I guess, but we have looked at several different families or several different offspring lines of Darling 58, and they vary a little bit in growth rates, very much like any unrelated populations of trees. So when we look at wild type American chestnuts from different parents, we see different growth rates. Some lines grow faster than others. And with the transgenic trees, we're seeing the same thing. There are differences between different families. The average growth rate for transgenic trees seems to be preliminary. Don't quote me, we're working on nailing this down. But the average growth rate seems to be a few percent slower than the average growth rate for non-transgenic trees. So certainly within the realm of the variability that we see from tree to tree. But there might be some differences, and that really makes a lot of sense. I mean, the tree is using energy to express this transgene, and so in any case where you have enhanced blight resistance, that's going to be a trade-off. The tree is using energy to deal with a pathogen, and that energy has to come from somewhere, and is not surprising that there are these small growth differences.
Great job, Andy, on the rapid fire questions. We're going to give you a break and flip this one over to Jared, but you're welcome to chime in. Finally, is there a way of feeling, is there a feeling of what might be the best trees to cross with Darling 58 to improve it? It seems to have an odd growth, so would fastest growing chestnut be best for crosses? Jared, you want to try to get that, and Andy can chime in.
So one of the things so there's like 400 million chestnuts estimated to be remaining on the landscape. So we're not going to be able to cross with all 400 million. So one of the things we're trying to figure out is how do we get a representative sample from all of the different climatic zones where chestnut grows? And how do we represent the diversity that remains in the species so that as climate warms, as we have, as we're trying to restore it to its large native range, that we represent all of the key diversity. So what we're doing is sequencing the genomes of 500 American chestnuts. And I appreciate the people that have helped with the leaf collection for this. And we're looking at how does that DNA variation correlate or is associated with climatic variation, and what are the zones of local adaptation in chestnut? Do we see, like, on a mountaintop, like, you know, top of the mountain versus the bottom of the mountain? Is there locally adapted trees, or is it much at a wider scale that's like more like from Maine, mid-Atlantic and southern region, like as we've kind of structured our breeding program? So how concerned do we have to be about local adaptation in terms of flowering time, growth rates? There tends to be, in the north, the trees that are more cold tolerant, they tend to grow slower, but they live in the cold. So they're able to tolerate the cold tolerance, which is Tom and Kendra have both done this work on this. So the short answer is we're trying to get a representative sample to cross with Darling 58 so that the tree can continue to evolve on its own in the forest.
Andy, do you have anything to add?
I guess just the part of the question about having an odd growth form, we haven't seen any differences in form. The only difference, like I talked about, might be growth rate, but that, like I said, that's within the variability that we already see, and it doesn't seem to be a different form. It's not, I don't know, growing shrubby like Chinese or something like that.
I forgot to mention one thing. In our breeding program, we have trees that are resistant to the second pathogen that affects, major pathogen that affects chestnut, which is Phytophthora cinnamomea, that kills the trees from the roots. And so we're identifying trees in our breeding program that have resistance to the root pathogen and crossing them with Darling 58. So we have trees that are resistant to the two major pathogens. And then we're also trying some crosses with our backcross trees that have partial resistance to blight to see if there's some kind of additive or complementary resistance when we cross the two types of trees together. So we're trying all of the different combinations, I guess, of, you know, wild type from all over the range, also backcross by transgenic trees with some with Phytophthora resistance and some with blight resistance to see which strategy ultimately leads to trees for restoration.
Great, and some really nice complimentary comments in the chat about from Anne from Paris. You've been a really great regular on the Chestnut Chat, and it's so cool that we get somebody from Paris. I took a lot of French in college, so I love I love thinking about that. Anyway, and Steve Johnston complimenting Sarah and her ability to facilitate and moderate all these. This really has become her baby, and she's done an incredible job. And there is a lot of competing content out there on Zoom and webinars, and everybody's trying to, you know, get on and get people's attention. And we've been really thrilled with the response to Chestnut Chat. So thank you. Now the $1 million question: distribution of seeds. When can I get seeds? When can I get plants? When can I get seedlings? How soon after the Darling will it be available? So I'll just kind of do an overview, and then I'll go through each of the questions one by one. As Tom mentioned earlier, we do have a seed sponsorship program for our best material. We have a $300 to $1,000 level of donation, and then you get seeds every year depending on the size of your donation. Every year it's kind of a numbers game, because as Jared alluded to, we are culling the low performers on the Meadowview Research Farm, which is the main area where we get our seeds from. So they're more rare than they used to be, because we do want to get down to the best of the best. But we do take a priority for the seed sponsorship donors, and that's been a really great way to help fund our research and our operations. So that's one way. I know there's distributions out there for Pure American Seeds And we have a Pure American program that we do every year. So the wild type Americans, I think we're getting away from the pure descriptor. And that's a great way to just practice planting chestnuts. And as Sarah said, yeah, if they get the blight one day, we'll just, replace them. But really getting pure Americans, wild type Americans in the ground is really important for the diversification and the projects going forward. So those are a couple of ways you can get seeds, and I will just throw it out to the group when I ask the questions. So thanks for all your interest. We get, probably all of us on the panels get these questions every week. And that just shows people are anxious and want to be part of this grand experiment. And we will do our best to be able to, accommodate those. So when are the trees going to be available? Can we plant in parks? Anybody want to take that one on? Kendra.
So as far as public plantings in places like parks, a lot of our chapters typically get a few of these, better selected nuts, just like our sponsor members or C-level members. And they do pursue demonstration and outreach plantings. So if you have a public space where, a few trees and some signage would help share the story of the American chestnut, I would encourage you to either contact Sarah, Tom, or myself as a regional science coordinator, or touch base with your local chapter and see what they may have available. Most of them are pretty amenable to those kinds of ideas, as long as, of course, we want the site to be appropriate for growing chestnut. If we put a demonstration planting in a bad chestnut spot, it's not going to look very good and the trees aren't going to do very well, but those are generally available.
Can I add one comment to the parks question? We're working, if you or your park is specifically interested in the transgenic chestnut, and if you're a little bit patient, we are kind of compiling a list of kind of educational partners or public plantings and things like that that might be interested in transgenic trees if they are approved with non-regulated status. So that'll probably be at least a little while yet, but feel free to reach out to me specifically and I can pass that along to our educational person.
Almost started talking with the mute on. I think we've covered most of these. There is one question about amounts of blight resistant trees. One person asked, can I get 200? I mean, we do have private landowners that are part of this experiment, but we have to really be careful in how we partner and partner smart, because these trees and these nuts are really first and foremost for our donors and for research. So we have to be very careful and hopefully keep the enthusiasm and the momentum going for people that want to plant it, but we have to be just kind of judicious as choosing who can be responsive. It's a big commitment, especially if you do a seed orchard. That's a 30-year commitment. So we just want to be sure people, we love the idea of backyard trees and ceremonial trees and demonstration trees to help, share the story of this hopeful mission. So we are definitely open to it, but it is limited how much we can accommodate right now. All right, so anything else on this? And we really don't sell them right now. There's some questions about sale. And that's just a long range, how we monetize all this, 40 plus years of research and development. That's a ongoing question. We're working with ESF on that. There is some intellectual property issues and questions involved. And we have a task force, again, with some really talented people, really great lawyer from San Francisco who's a plant patent expert, but we will not be patenting it for the time being anyway. There's talk about trademark and so forth. So I hope I covered most of the questions. Throw them in the Q&A if I glossed over something, but there was like maybe a dozen and a half of questions just on distribution. Anything else I might have missed, panelists?
I just wanted to mention that part of this relates to, okay, so if you're talking about when are you going to get a transgenic tree pending deregulation, part of this relates to like how many trees do we have in the ground right now that are transgenic and what are the stages of growth. because they would become the parents that we would generate seeds to distribute to people. And right now, our oldest plantings are at SUNY ESF, and there's like a few hundred trees, so we're not talking about huge numbers already. We are doing some crosses and planting orchards, but then it takes a while for the trees to grow up. So like, up to 7 to 10 years to get like large numbers of seeds off of these trees. And then, and that's just with doing controlled pollinations. So if we wanted to, this like kind of an exponential math to this. You start off with small numbers from our controlled pollinations, but then as we end up doing more crosses and put in more of these orchards and they grow up and they start producing more seeds, and we can plant more orchards and we can produce more seeds, so right now it's just limited in the sense that we have a very few number of founder trees and they're not grown up to produce large numbers of seeds. So if you wait a little bit, the trees got to grow, it's not, we're trying our best to make it go fast, but so the trees, they're going slow.
Thank you, Jared. Okay, we're going to switch gears here and talk about the breeding program progress, to Jared from Bill Loftus and Eric Massant. What is the current success of the breeding program? When did it start? How big are the biggest trees? I'll try to just stop there. There's several questions in a row. What is the current success of the breeding program? And I can tell you it started back in 1986 when Fred Hubbard started at the Meadowview Research Farm. Take it away, Jared.
So what we have is at Meadowview, we planted in our current generation seed orchards, we planted trees, like 60,000 trees. And we aim to select around 1% of those trees as the best performers to advance in the breeding program, like to get seeds from, to distribute to people, to plant the next generation, etc. So we are finishing selection. We basically finished selection in one of those orchards last year. Dan and crew at the farm did, and they're currently culling out additional inferior trees from those orchards this year. And so we'll have mostly done with selection after we started planting those orchards in 2002, and it's 2020. So it's taken us 18 years to plant those orchards and to complete selection. And what do we get in the end? We get trees with this spectrum of resistance. So there are trees that have more Chinese ancestry that have higher levels of resistance. We have trees that are more American that have partial resistance, but they are on average, the trees are about as resistant as the intermediate resistance of Chinese and American hybrids. So they get the blight, they have like cankers up and down the stem, but they're resisting the blight, they're able to survive. The trees that we've had in those orchards have survived for multiple decades. Some of them are not pretty, but they do live. And as we continue to call that orchard out, we improve the resistance incrementally. And as we plant the next generation and we do more selection, so the program has not yielded what we thought, which would be Chinese levels of resistance, but it has yielded resistance above American chestnut and overperforming relative to the amount of, so the trees are like 80 to 90% American chestnut ancestry, but they have the resistance of the 50-50 hybrids. So they're still doing pretty good for that. So yes, it's a slow process to do this breeding and to do selection and to call out a lot of trees, but we're speeding it up with genomic testing. And so the next generation of trees, we select the best parents, and we use DNA testing to make early selections, like 5 years. So then we're talking like kind of similar time frame for the darling trees, where we've made gains in resistance, in the next 5 to 7 years. So, yeah.
Thanks, Jared. And shout out to the team at Meadowview Research Farms, the 5 talented folks there, Dan, Jared, Brandon, Jim, Lily, and Eric. They're just really dedicated folks, and they do a lot of hard physical labor, which is involved in culling these trees, and they do a lot of really interesting science. So When we open up again, open invitation to visit Meadowview with advance notice, please. Jared, how big are the trees there? How big are the biggest trees? And do they look like American chestnuts?
Yeah, the biggest trees are like 30, 40 feet tall at this point in the seed orchards. And yes, they look like American chestnuts in terms of the leaf morphology. We're actually looking at the architecture, the growth form of the trees right now. So we want to know, does having 25% American or Chinese chestnut ancestry, or maybe 20% or something, make the trees grow shorter, I guess, and they have more branches coming off the side than the typical American chestnut. So we're looking at the size of these side branches. American chestnut tends to put more growth into the main stem and grow taller on that main stem, whereas the Chinese chestnuts tend to put more resources into these big side branches. So they grow like sprawling more and shorter. And so we're just kind of looking at trees that vary in the American and Chinese ancestry, like what is that growth form and how does it correlate with the ancestry of these trees. So that's something that's ongoing. We want to see, we want to kind of know, are these trees going to grow to the canopy? That's a big question. It's hard to answer because it takes a long time for the trees to grow.
All right, I think you've covered most of Bill and Eric's questions, but Roger Wilby from Maine asks, should we plant small plantations, not large ones, which attract disease and have natural selection take over and improve resistance in these populations? You've already addressed his intermediate resistance to the blight question, so.
Well, I'm kind of thinking, the way I've been thinking with all this genomic research is, if we can select the best parents and we remove some of these trees that have very little resistance or inferior, that dragging the population down, I guess, in terms of resistance, and we do a better job of selection, so quality over quantity is kind of where I'm aiming with the breeding program. And in Maine, we could select some of your better parents, and you could put out smaller plantations based on with your better parents out in the forest, and continue the breeding program in a way in the forest, naturally allowing natural selection to select the best kids from those parents. But the parents will be better on average, so their kids will also be better on average. In terms of where we do with our orchards, we might not need to plant out as many trees, but we do a more careful job with our selection, inoculate, wait a little bit longer to make our assessments, do the genomic testing, make really good selections. I think that's where I'd like to see us go, rather than plant out hundreds of thousands of trees that are a lot of them inferior and then having to cut them all down. It's a lot of work.
Thanks, Jared. So process check, we're about 20 minutes to 1. I can stay on as long as everybody else can. We're probably a little more than halfway through the questions, believe it or not. So maybe we can be more concise in our answers, panelists, just so we can get to everybody's questions. So we're going to turn it over to wild type American chestnuts. And I'm going to flip this one to Tom, John, and Larry from New Hampshire and Massachusetts. They have American chestnuts in their neighborhoods. How do they find more? And what should someone do if they find a wild tree? Anything that should be done to protect them.
Okay, so you have chestnuts in your neighborhood, presuming that they're naturally growing, that means you're in the chestnut range. If people want to find American chestnuts, you should look at a map of the chestnut range, like the little map, and look for places where you're going to be in the natural range of American chestnut. You don't want to be out in the Piedmont, for instance. You're probably not going to find them there in sites with clay soils. And then once you're in the chestnut range, depending on where in the range you are, you can think of a couple of these tips. If you're in the south of the chestnut range, chances are you want to get higher up in elevation. Chestnuts tend to grow on the hilltops or in the Appalachian Mountains in the south. As you move north, you're going to find them at different elevations, but again, you're still looking for the hilly country because chestnuts are an upland species. They like to grow in the hills. And as you move up into New England, You're probably not going to be on the tops of the mountains because chestnuts aren't adapted to that climate. You're going to come down from the mountains, but you're still going to be looking for the hilly country. And the reason why you're looking for hills is because chestnuts like moist but well-drained soils. And therefore, you don't want to be down in bottomland sites. You don't want to be where soils are clay or wet, or or where where the soils don't drain very well. So you don't have to go out in the woods and start doing soil tests. Simply do this: go to a place where you're going to find a hardwood forest, not those sites where there's carpets of ferns and where you find a lot of birches and where the soils are damp, but move upslope to where you're going to find a lot of other oaks. One of the most common occurring trees that you'll find in sites where chestnuts do well are chestnut oaks. Their ranges overlap and they seem to like similar types of environments. But any oak hickory forest is going to work if you're in the chestnut range and you're in a nice oak hickory forest. You're probably in the right soil type. And I would say those are good places to search for chestnuts. There's more information that we published in, I believe, the last journal, or maybe it was the one before that, the summer journal, where we discussed looking for American chestnuts. And so that would be a good place to look for a little bit more information on how to find them and tips for going out and knowing where to look. If you find American chestnuts, we ask that you collect data on them using the TreeSnap, using the phone app called TreeSnap. So any mobile device, go to your app store and download TreeSnap. Once you get TreeSnap, you can click on American chestnut and it allows you to take pictures of the chestnuts that you found. We would like you to take a lot of pictures, especially of the fronts and backs of the leaves, so that it helps us identify them. We would, of course, love if you collected a leaf and sent it to your regional science coordinator, but just getting a lot of pictures, looking at the leaves and looking at the site where you found it could help us know whether or not it's really an American chestnut. And then you collect some data and it automatically uploads your GPS points, so we'll know where the tree is. And this will work even if you're not in cell phone service. It'll collect the data, and when you get back in service, you can upload it, the identification, and then we'll know, and we'll be able to go back and look at it. We'll work with our members to maybe go out and work with those trees. There's nothing you really need to do to protect the trees in the wild. There's a lot of trees out there, and they're probably little stump sprouts. Maybe they're big trees, but I'm not going to recommend that you do anything to save the trees. If it's on the woods in your property and you want to put a deer cage around it, deer will eat these wild trees. We see it a lot. They especially love to go after the stump sprouts. You can protect it if you want from herbivory, but I wouldn't say that's really necessary. So just go and find lots of trees, and everybody should be out there hiking around in the woods this summer and find trees and then record them in TreeSnap.
Thanks, Tom. And a friendly reminder, let's keep our answers as concise as possible. Tom is so full of information that he can't help himself. Thank you, Tom. I'm just picking on you. This is to Jared. In regard to disease resistance, has useful information been obtained from GWAS, which is Genome-Wide Association Study, for those of you that don't know, and I didn't know before I started this job, GWAS examination of the back-cross American chestnut trees?
Thanks, it's a great question. Close to my heart. We have found regions on nine chromosomes out of 12 that are associated with resistance. Some have larger effects than others, but it does not look like there's any major large effect genes. It looks like they're scattered about and they have small to moderate effects. So the back-cross program was based on the idea of like a few genes, and that does not seem to be held up or supported by the GWAS.
And is it feasible to catalog genes lost during back-cross, which most frequently coincide with the loss of disease resistance?
Yes, we have a way of basically knowing where on each of our back-cross trees we can say, in this part of the chromosome, it inherited its genome from Chinese chestnut, and this part it inherited from American chestnut. We kind of look at the mosaic of inheritance and how does that compare to where we find regions that are associated with resistance. We can say this tree has this region A, B, and C that are associated with resistance, but it lacks D, E, and F. So yes, we can do that. We don't know the exact genes, but we know big regions of the genome that seem to be correlated with resistance.
Thanks, Jared. And those two good questions were from Tom Obrick. Thank you, Tom. Back to you, Jared, from Mimai and Thomas Morgan. What is diversity like in the wild type American chestnut? Are there known niches or pockets of diversity like in uplands or lowlands, or any known subspecies? And what are TACF and partners doing to capture diversity and diversify resistant trees? So if you need me to repeat any of those, let me know.
Okay, so the sequencing original of the 500 American chestnuts, we find we find that there's a DNA variant every, the chestnut genome has 500 or 700 million bases in its genome. And we found that there's like 45 million of those sites in the genome are variant between species. So there's lots of variation in chestnut, and it looks like the South seems to have the highest level of diversity, particularly in Alabama. It has like, even though there's not that many trees, there seems to be this weird subpopulation in Alabama. And it might be due to the chinkapins crossing with chestnuts. I want to know that. And so we're sequencing a bunch of chinkapins, which are related, they're cousins of chestnuts. And we're seeing, did the chinkapins cross with chestnuts and give some of their DNA to contribute to that resistance that enable them to live in the hot red clay of Alabama. So we're prioritizing the southern regions, particularly over by like, Western Tennessee and Alabama and Mississippi to conserve that germplasm, because climate is changing and these trees are not so happy down in that particular environment. That seems to be the highest levels of diversity, but we're still doing the analysis.
Sorry, I was managing chats and texts here. Let me see where I ended up. Thanks, Jared. This is for Kendra from Frank Skalak in Ohio. I have seen after a chestnut dies back from blight, some trees will send up a large number of root sprouts, while others will send up only one or two, and sadly, sometimes none. Is this strictly controlled by genetics, or does environment play a role?
Well, I think this is a great question, and I've often found that chestnut folks are wonderful at asking questions that no one else has really put a whole lot of thought into. I have several volunteers that I visit when I travel around the region, and often over the dinner table, they ask me questions. I'm like, I don't know if anyone has ever looked at that. And I think this falls into that category, Frank. I'm not sure. My general impression is, and I actually did spend a little time looking into, what controls chestnut or, trees to resprout. I mean, it's a stress response. I think we're all pretty aware of that. Most of what I could find, in a quick Google search is people trying to prevent it from happening, which is not what we want. But, my impression with chestnut is that it does resprout as a response to blight, but how many times can it do that? When a mature tree dies, there's an awful lot of energy stored in that root system. And so those adventitious buds that are all around the base, kind of sitting in wait and ready to sprout, have a lot of energy stored up to do that. And as the tree goes through successive diebacks, it runs out of energy. And so I think that's generally what's causing, different numbers of sprouts to come up, and certainly smaller numbers, or sadly none, is that just the tree runs out of steam. If any of my panelists disagree with that or have anything to add, I welcome that, but I know we're a little short on time. That's my best explanation for you. Thanks,
Kendra. So since we did run out of time, we hate to not answer all the questions today, but I think I'm going to suggest that we be sure that we capture the questions that were submitted. We can do some email work to get back to all of you, but just a reminder that the Chestnut Chat in February is going to be on planting. Is that right, Sarah? Planting and
No, so okay. January is going to be biocontrol. I had a lot of people ask about biocontrol hypervirulence, so we're going to have Amy Metheny come in from WVU and talk about hypervirulence. February is going to be on historic importations and cultivars. So we'll talk a little bit more about Dunstan and other things. I'm super excited to invite Sandy Anagnostakis, Dr. A, and also Michael Nave from California. He is like the world's most knowledgeable guy about planting. He's got everything. I mean, everything you can imagine he's got in his backyard. So that's February. March, we were going to talk about chestnut controversies. So if you can think of a controversy within the chestnut world, let us know. I think we've come up with enough to talk about them. We may or may not, so that's tentative. I do love the idea about chestnut controversies, but because a lot of those questions, Lisa, that we're not able to get to are about planting and growing, we might want to switch that out. We might want to talk about planting and growing. March is the perfect time to do that. So I think we'll either have a special planting session on one of those other Fridays, or we'll replace chestnut controversy. But we won't let the controversies go. We will definitely talk about them. It just may not be in March. And then April, we're going to talk about biocontrol again. I got Mark Double coming in to talk more about cruddy bark, to talk about how cankers change over time, and also Laurel Rogers from Shenandoah. She's done shotgun sequencing of cankers. And so she basically takes DNA from the cankers, throws them through a sequencer, and then figures out what the heck's in there. Bacteria, fungus, you know, all kinds of stuff is growing in that thing. It's kind of crazy. So that's April. And then we haven't gotten to May yet, tentative. And then the rest of the time we got to figure out. But I think if you have any ideas for what you want to see out of this, any topics, you know, let us know. That's really helpful. Or not just topics, but people. Is there someone that you'd want to hear from in the chestnut or beyond community? That'd be helpful, and I can line them up. We've got 6 or 7 left yet to go. Well, maybe 4, because I got some ideas for other ones, but I would love to hear your ideas of what you'd like to see.
So with that, any last other remarks from the panelists, something that you meant to share and didn't and popped into your head?
Yes, because I like to
I
talk
had
a
one
lot.
too, Tom. You
Okay,
go first,
Kendra and Tom, you
Yep.
got 1 minute each.
Okay, I was just going to let everybody know that a lot of the information that we talked about and some of the questions that we didn't get to, there's answers on our website. So go to our website, you'll find a lot of information there. You can also go to our website to find fact sheets. So if you're interested in, say, dealing with ambrosia beetles, there's a great fact sheet about that. Harvesting chestnuts, all those things. There's a lot of information on our website, including on our fact sheet. So go there. If we didn't answer your question today, you can find the answer on our website.
Tom, good job.
And I
Very
just wanted
concise.
to give a shout out. Thanks, Tom. Barbara Evans, you had sent in a question ahead of time and asked the same question in the Q&A, and we said we would answer it live, and we failed to get to it. And it was one that was assigned to me. So I just wanted to let you know, I put a whole bunch of links on seed stratification, harvest, and storage in the Q&A. If you have a chance to grab those, please do. If not, shoot me an email or I'll try to follow up with you offline. But I just wanted to let you know to check that out. There is some info for you.
And you meant the chat, right? Not the
Nope.
Q&A.
Nope. It's
Oh,
in
the Q&A? Okay,
Yep.
very
I
good.
added an answer to it.
Very good. And also on our website, before we hang up, it is our most important fundraising time of the year. So if you can spare a few dimes for TACF, believe it or not, we're running ahead of last year's fundraising. This end of year campaign is our most important. We get 94% of our revenue from private individuals, family foundations. We don't get much government support except from the Forest Service. They've been wonderful for so many years. But please, if you consider us in your end of year charitable donations, we'd be very, very grateful. And I just want to thank this wonderful panel. They are just the most dedicated, wonderful colleagues. And with that, I hope you all have a safe and happy holiday. Let's get through this nasty old year of 2020 and get on to 2021. And thank you for all you do. And with that, I'll hang up. Thanks, everybody.
Thanks, everyone.
Take care.
Bye.
Bye-bye.